Hi, my name is Phil Ewels and I work at the National Genomics Infrastructure, part of SciLifeLab in Sweden. We work with research groups from right across Sweden who come to us with their samples. We do library preparation if required, we do sequencing, and we also do some limited bioinformatics work. We process a great number of projects and samples every year, and quality control is really important, and so this talk will be about how we handle the high throughput quality control in our facility. The Stockholm branch of NGI is an ISO accredited facility where I work. That's for the library preparation in the lab, the sequencing, and also parts of the bioinformatics analysis. According to the reports that we send to our users, this means that all the data is subject to highly stringent quality control procedures. But what does that actually mean? To give you some context, we have quite a lot of sequencing machines in our facility, and we process very many thousands of samples. So in 2016, we had over 12,000 RNA samples, for instance. The amount of sequencing we actually do is equivalent to kind of 1x human genome every 4 minutes or so. So it's a vast quantity of raw data that comes out of the facility. And we have to handle quality control on all of that before delivering to users. This quality control at scale is essential, we think, and really helps us to catch things early on, which hopefully saves a lot of time both for us and also for users later on. And we approach this quality control in a number of different ways, and I've laid that out here as to how kind of the frequency of these checks. So on the left, things we always do for every single sample, kind of automation and visualization. And over on the right, the more sporadic things that we do less frequently, more in depth. I'll start off with the kind of automation side of things and how we handle quality control for every project and every sample. So we in order to power the automation of especially our bioinformatics, but also in the lab, we have a database to keep all the information about every project, which we call genomic status in-house. They have different sources of information feeding into this database. So when research groups want to start a new project, they do that through a website we have, which we call the order portal, which collects all the initial information about that project and puts it into our database. We manage all communication with users via email through the Zendesk system, which also ties into genomic status, so we can easily hook up that information and link to specific projects. And in the lab, we use the Genomics, or now Illumina, Clarity LIM system, where we track all information about different reagents and everything to do with the project. And that feeds, again, into genomic status. We basically copy the information out of the Clarity LIMS database into genomic status, so that we have everything in one place. We do all of our bioinformatics work using automated workflows. So that has been a tool called Piper in the past. More and more we're moving to using Nextflow to run all of our different analysis pipelines. Coupled to these pipelines is an automated collection of results and with that visualization of those results. We use MultiQC, which was a tool that I wrote for this purpose in-house, which has been really good and has been well welcomed by the community. And that passes pretty much any different type of log file that you can throw at it and visualizes it so that it's easy to understand. We actually use this in tandem with a couple of different plugins that we have for MultiQC. So there's a plugin which is available for anyone else to use as well, which talks to the Clarity LIMS system and pulls in metadata from the lab into our reports, so like the RIN score of RNA samples or the loading concentration. And we have a specific NGI plugin which then pushes information passed by MultiQC from the bioinformatics results into genomic status, and also saves reports to our web server so we can easily view them without having to go onto the analysis server. Genomic status itself in front of the database is also a website, and this is where we usually go to find out information about projects and keep track of everything that's going on. This is a typical page showing a specific project in our system, and you can see it's ongoing, and we have all the key kind of timestamps for the different stages of the project. We also keep lots of different information, but most of this comes from LIMS, but also the order portal. So you can see everything about the project pretty much at a glance. These different tabs then show us more information about specific things. So the samples tab shows us each sample which is in that project, and shows us the validation, who did it, different statistics, and links through to statistics from that flow cell as well. And you can see there's a tab for user communication which shows the Zendesk tickets, and we can make notes under running notes and various other things. There's also a bioinformatics QC system built into the genomics datas, where for every sample that we process, we go through this basically series of checklists for each sample, where we check the qualities of those samples. There's also a section of this which is dedicated to the specific type of project it is. So here, this is a whole genome resequencing project, and so we check the coverage and duplication for every sample. We're currently writing a plugin for MultiQC which will automatically set some of these values. So for an RNA sample, it will automatically put in whether the alignment rate was good enough for a pass or not. Whether this is a pass, warning or fail is something which we have decided early on and is standardized and written into our documentation systems which we handle with AM systems. So this is consistent for every single project we run and basically signed off. We can also easily get to the MultiQC report from this project, which is all automatically synced by the pipeline and by MultiQC. And a MultiQC report, for those of you who haven't seen one, looks something like this. So we kind of have some information at the top about the specific project, and then lots of information contained within this report. At the top you can see every different sample and some key statistics, and then there's lots of different plots which are visualizing quality control metrics from this project. This makes it really easy to spot outliers. Here we can see that one of the samples has a low quality, so I've highlighted it, and we can go back through the rest of the report and see if there's anything else that looks weird with this sample. Sure enough, you can see that the quality control and the sequence content is a bit different with this sample. So this allows us to spot problematic samples even in very large projects very quickly, which is really helpful. So that was all the high throughput checks that we do kind of routinely for every sample, but we also have more occasional quality control steps that we take. For looking at longer term trends, a project that we're working on at the moment, which tentatively called MegaQC, but then the name may change, I'm not sure if I like it yet. And basically the idea with this is we collect lots of really interesting information from MultiQC, but it's always for a single project. And so the idea for this is to be able to collect information from across all MultiQC runs in a single database and then visualize that in a website. So this is the mock-up that I've put together, which we're working on, and basically be able to choose a different type of plot, so either a plot from within the reports or pick values, and so I can pick a trend in the specific values here, pick out the samples that we're interested in, so filter for different kinds of fields, and then plot that. And hopefully this will allow really fast and intuitive visualization of different quality control metrics so that we can spot things easy in large trends. That will be publicly available when it's finished, but we're just starting on it now. Something else we do routinely, especially more on the lab side of things, but also on bioinformatics, is what we call validations. And these are really in-depth reproducibility checks. So every lab technique has a protocol in our documentation system, and if we want to make any changes, we have to rerun a validation. This applies to the bioinformatics as well. So every time we make a new release of an analysis pipeline, we also have to rerun a validation. In addition to those validations, we also do audits. So we do horizontal audits where we check a single step in our workflow for lots of different samples. We also do vertical audits where we follow a single project or a single sample from kind of initial submission right the way through to delivery of data. We have multiple branches of NGI, so we have a sister branch in Uppsala, and we often do these audits with them, and we can check the reproducibility of the data that we generate with them, which is very helpful. And we have also worked with other sequencing facilities across Europe. So for the bioinformatics side of things, I said that we do pipeline versioning of each workflow, and that we rerun validations every time we make a change. And every run is logged and reported, so it generates an HTML report saying exactly what version of software was used and whether there was any errors, and that's saved both with the project and is also emailed to our bioinformaticians. pipelines themselves are hosted on GitHub with the help of software packaging in Docker, which is facilitated by Nextflow. It means every time we push a single change to the code, the pipeline runs with some example data on Travis, and we are immediately notified if we have broken anything inadvertently. This is really, really helpful and gives us confidence in our code when we're making changes. I said that reports are generated from the pipelines, and this is an example of one such report. So this is both saved in the analysis directory and also emailed to us when we're running the pipeline. And you can see at a glance that everything has worked quickly by the happy green status at the top. But we also log the exact command that was used to launch the pipeline, where the input data files were, all the different parameters we used when we were running the pipeline, as well as software versions and lots of other information, which is helpful if someone asks in 2 or 3 years' time when they're writing a paper, what version of star did we use to run this alignment, or whatever else. This is all handled through Nextflow, which we're starting to use more and more, and is really turning out to be an excellent tool for this. It's really, really helped us. And we've got several other pipelines that we've written in addition to this RNA-seq pipeline. So small RNA-seq, methylation-seq for bisulfite data with Bismarck, and ChIP-seq pipeline. And we've got more on the way as well. So that's everything. If you're interested in any of the software we've written, we have a website called opensource.sciliflab.se, and that lists all the different packages that we've released, which are available for others to use. I talked a bit about MultiQC, and that has its own website, so multiqc.info, where there's lots of documentation and examples where you can find out more. And all of the code that we write, even the developmental code, is all kept on GitHub, so you can find all of that under our organisation, which is called SciLifeLab. I'd like to say thanks to Max Keller, my boss, who's allowed me to work on all this stuff, and other members of my group in the applications development group. And feel free to get in touch by email, GitHub or Twitter or anything else if you have any questions. Great, thanks very much.